Okay, so this is the new book. It launched in the U.S. in February, and this is the U.K. book launch now. I'm here. I was here for a week, and this is the only animal for which the entire connectome has been mapped. You guys are familiar with C. elegans. This is the favorite worm studied by biologists. It's a millimeter long, and this is its connectome. So what my joke is this reminds me of the back pages of airline magazines. Somebody said it looked like the London Tube. I always ask my audiences what it reminds them of. But yeah, so if you take an airline map, replace every city by a neuron, and every flight between cities by connection, you get a connectome. More formally, a connectome would be a directed graph, because the connections between the synapses between neurons are chemical synapses, at least, are directional. So A sends a synapse to B, and not necessarily vice versa. Yep.
Is edge length meaningful?
There's a graph layout problem here, and it doesn't have a direct meaning here.
Okay.
So there's some algorithm that was used to lay out this graph, which I'm not going to talk about. So your connectome then would be 100 billion nodes and roughly thousands of edges per node. So a lot more complicated than this. And so mapping out connectomes is a big technological challenge, because even this connectome took over a dozen years to map in the 70s and 80s. And so what's been happening in neuroscience is that this was so traumatic an experience that nobody wanted to do it again for a long time. And the technologies have gotten better, so now we're dreaming of taking improved versions of these technologies and applying them to brains more like our own. And I will just say, so I'm going to talk about the technology first. Well, this is maybe not the best, well, maybe not. Let's talk about the motivation. So there's many motivations for studying connectomes. I would... Well, let's see. Should I talk about that? No, I'll go on. Let's just talk about the technologies. So here's a piece of mouse brain. It's embedded in a hard plastic resin. It's been stained with heavy metals. And here is a very high-tech deli slicer. So this is a blue tub filled with water. That's a diamond knife. And here is a piece of brain inside that plastic. And it goes back and forth. Really thin slices. Say 30 to 50 nanometers thick, really thin slices, float onto the surface of the water. They spread out flat. And then here is a conveyor belt which collects those slices automatically with no human intervention. And this is a very old technology, this slicing technology. Ken Hayworth has recently added this tape pickup to improve reliability, because previously people would fish the slices out by hand. So you slice them really thin. You mount the strips of tape on an electron microscope. This is based on images from the laboratory of Jeff Lichtman at Harvard, where Ken had been working until recently. And we're zooming in by a factor of 100,000 times until we get to this resolution required to see the wiring of the brain. And we can take images of many slices and stack them together to make a three-dimensional image. And then now we can color in the cross-sections of one branch of a neuron, slice by slice, over and over again. And by doing so, trace the path of that wire of the brain through this volume. And you can see that just as every rose has thorns, a neuron has thorns too on its dendrites. They're sticking out. This green neuron is touching the red neuron in two locations. And if you look inside one of those locations, you can see little round circles. Those are vesicles. They contain neurotransmitter. So when the green neuron wants to communicate, to send a message to the red neuron, it spits the contents of those vesicles into the cleft between them, and then the red neuron senses those molecules. So there is do you philosophers, have you heard of Pierre Cabanis? French philosopher who wrote, "As the liver secretes bile, the brain secretes thoughts." And it's sobering to think that when you're thinking the most refined thoughts of philosophy or love, your neurons are spitting on each other. So these images demonstrate that if we have really high resolution images, we can follow the wires of the brain, we can find the synapses, and therefore we should be able to find the wiring diagram or find the connectome. You guys convinced of that? The catch, of course, is that requires doing a lot of image analysis, and it's not enough just to fill in, if you want the whole connectome, it's not enough to trace one or two wires, but you've got to trace all the wires inside here, which is kind of like coloring a three-dimensional coloring book. And I'll show you what those look like. So brain tissue is packed. You've normally seen pictures of neurons, one neuron alone, and that's through a trick. You stain one neuron and you leave the other ones transparent, and then one neuron is revealed. That's the standard way of imaging neurons. But with the electron microscope, we can see all of them at the same time, and now you can see that it's just packed in there. Now, this was manually colored in by hand by a postdoc in my lab named Daniel Berger, and it took him 250 hours of work. And I can guarantee you that Daniel is much better at this than you or I. He is almost superhuman in his ability to do this coloring task. He's extremely accurate and he never gets tired. And that's a tiny piece. That's 6 microns on a side. So that's how that piece stacks up against an entire neuron. And that's how that neuron is compared to a brain. And that's just a mouse brain. So you can see from this that finding a human connectome would be it's an incredible challenge, and a lot of it is getting the images, but a lot of it is just the image analysis. Actually, that little cube didn't take that long to image, but it took 250 hours to analyze. All right, so that's the scale of complexity, and you know, people sometimes say the brain is the most complex object in the universe. Journalists love that. It's kind of this piece of hyperbole. It's not really clear what it means. But here you see the material complexity of the brain's wiring. I estimate millions of miles of wiring inside a brain. Okay, so, and I guess I should show you this other thing. I'll show you. What should I show you? So here's a technology that's going to be ready supposedly by the end of 2014. I visited Carl Zeiss Incorporated in Germany. This is the mother of all microscopes. It's 61 scanning electron microscopes in one. So 61 beams scanning in parallel. It's going to generate 1 gigabyte per second or 1 petabyte every 2 weeks. So that's a data rate higher than that of the Large Hadron Collider. The first 2 models, commercial models, will be delivered at the end of 2014. And 1 petabyte, at what you just saw, if we imaged at that resolution, 1 petabyte would be 1 cubic millimeter of brain tissue. Not a whole brain, but a little piece. And that contains 100,000 neurons and 1 billion connections. And just remember that MRI blurs all that into 1 pixel. So there's a huge resolution difference. And if we extrapolate, Daniel coloring this would take him probably 100,000 years if he never slept or took coffee breaks. And we built AI to speed up the process, but it's not very accurate. All it can do is speed up humans. So if humans interact with the AI, then maybe they can go optimistically, at this point in time, 100 times faster. So it's still 1,000 person years. And Okay, so where are we going to get that? And I could show you a little bit. If we have time later on, I'll show you what the real data looks like, but more of that. But we've worked on AI for quite a while, but these days we're really working on social computing. And so my answer for where we're going to get this time from, this labor from, is by copying these guys. So does anyone know how much time the human race spends on Angry Birds every day? 600 years per day. So 2 days of Angry Birds, in principle, would be enough to completely find all connections inside a petabyte or a cubic millimeter piece of brain. And 1% of the labor in Angry Birds would enable us to do it in a year. So, although getting 1% as fun as Angry Birds is still not easy. And I will show you. Let's see, do we have anything here? Okay, we are logged in. So we are now, in the last year and a half, I've been putting a lot of effort into citizen science. So here's the first citizen science site for connectomics. You've heard of Galaxy Zoo? Galaxy Zoo? So astronomers have mobilized volunteers to help out in analyzing galaxy images, and Foldit is an example of getting people to do, amateurs to do protein folding. So we're trying to get volunteers to help us with neuroscience. So there's some educational material here about neurons and about the retina and so on. There's a call to arms. Welcome to Eyewire, where you can help make discoveries about the neural structure of the retina. This was written by an undergraduate named Elizabeth. I had a short class in January where students helped us create this website, and Elizabeth wrote this one. She walked in one morning with a piece of paper, and she read this out loud. She read that. She wrote, "To achieve this, we need something more intelligent than even the most powerful supercomputer, you." And then students spontaneously started applauding her. I've never seen that in all my years of teaching at MIT. It was like the high point of teaching. All the students applauded one student spontaneously in the middle of class. There is, and here's the game. So there's 5 players currently online, and you can see here, this is a neuron that they're currently reconstructing. It's one of the neurons in a data set of retinal images. There are green boxes here. Those are the locations of those players in the image. Each of those boxes is like the cube you saw before. And there's a leaderboard, so they get points. Each of those green boxes is a task where they trace a wire through it. And there's a leaderboard because they get points after each task, and so they can compete with each other. So if you're a new user, you sign in, and you see an instructional video, which we'll skip. So this is a cube of the retina, and the location of this plane shows the height of this slice inside that cube. So you can see that, and we can move up and down, depending on how fast our internet connection is. Okay, so you can see here that the AI has colored in part of a branch. It stopped when it's not sure, and you can see that that's rendered in 3D on the left, right? So your job is pretty simple. It's just to click and instruct the AI where to go on. So if we click here, the AI goes on. So it's filled in the rest of the branch, and that has appeared on the left. And it's done that in 3D. So it's a man-machine interaction. And these are tutorial tasks in the beginning. You're learning to be an amateur neuroanatomist. If you click in the wrong place, then it turns red, because an expert has already done this. So you're being compared with an expert. So there's 15 tasks. They get harder and harder to find the missing branch. And when you're done, you graduate and you're doing the tasks for which no expert opinion is available, and you don't get any feedback like this. All you get is a score. The score compares you with multiple people who have done the same task. And if you agree with other people, then you get a higher score. There's potential pitfalls for that scheme, but it sort of seems to work. Okay, so that's the basic game. And the first interesting empirical result is that some people really like this. Some people get addicted. And there's even at this, it's an amateurish game, but it's engaging. And I think part of it is that this is kind of mesmerizing. It's like watching the clouds, right? Organic forms shifting. Some people compare it to a lava lamp. There's a certain kind of pleasure that comes from completion, of finding places that are not done yet and filling them in. So that's kind of the crossword puzzle pleasure of completing something, filling in the boxes. And then there's the feeling of competition on the leaderboard, of getting better and better over time, stuff like that. But we found out more about our users on the forum. So we made this, and so my joke is that I knew we were onto something because one night I asked my girlfriend a question, and she said, "Don't bother me, I'm finishing a neuron." And the other thing I learned about my girlfriend is she seems really mild-mannered, but she's quite competitive. And she was obsessed with being number one on the leaderboard, and so every night she would do enough tasks to get to number one, and then she would be triumphant. But every morning she would wake up, and Jose was number one. And this went on for weeks. And at one point, she just expressed her frustration and said, "Honey, can't you do something about this Jose guy?" It's like putting some stress on my relationship, and I stood firm. I didn't do anything. I didn't do anything. But we eventually found out that Jose, because we had this forum, he was one of the first people to post extensively on the forum because he's an avid user, and Jose lives in Spain. And so the reason Jose was number one every morning is that he woke up hours before, and he was at work, and so he would become number one. It's an international effort, the number one shifts. And so he said, "Hello, fellow Eyewire." So they call themselves Eyewirers. And he says, "I play with my 7-year-old daughter." It's like, wow, it's a family activity. I didn't really think about kids playing this. And then he said, "And look, we found this piece of a neuron. It looks like an ice skater." And then I gave you the finger. So the users started running with this and having a good time, right? So we were really stunned by this because it was something that we never expected. They were being creative in a way that we never expected. It was wow, neuroscience is fun. This was really really exciting to us that people were having a good time. And we also had some more serious kinds of contributions. Like I was complaining to my programmers for several weeks that the performance of loading images was bad sometimes. And they said, "It's your problem. Your home internet connection has long latency packets, has drop packets. It's your fault. We can't do anything." And then one morning, this guy says he notices the same problem, long load time problems. He diagnoses the problem. Eyewire isn't making use of HTTP pipelining at all. I don't know what that means. And then he gives the fix. You look into using a different proxy solution for the front end. Use the keep alive header. Just my 2 cents. And the programmers changed 3 lines. Problem solved. they were really embarrassed, but they were also, we were elated that somebody had just showed up on the discussion forum and fixed our problem, right? So I become convinced by this experience that citizen science is going to be crucial for driving forward the technologies of connectomics. So right now, my major activity is that I'm trying to find all the partners that I need to build a venture that breaks down the barriers between research, education, and entertainment. That we can discover things about the brain and learn about, people can learn about the brain. They can also be entertained while they're doing it. So why is this crucial? Some people say that we need. What can I do here? Oops. Well, what am I doing? Well, okay. So why is this crucial? So the reason is that I believe to get technological. So my goal here is we need to get to the point where we have exponential innovation in connectomics if we ever want to succeed. I showed you that huge gap between what we can analyze now and what an entire brain is. The only way we'll ever do an entire brain in my lifetime would be if we had exponential growth. That every year we got 2 times faster or 3 times faster at doing the image analysis. So how do we get to that point? Well, I think we need to have both crowds and machines. We have to have lots of people who are taking the inaccurate output from the AI and fixing it so that we can generate science now, so that people actually care. We can't say, 50 years later we'll have science. We got to get some science out of it now. And then, at the same time, the crowd is teaching the machines to be smarter, because the AI is trained by machine learning. And having a large amount of human expertise feeding into our AI will make it faster next year. And to create all this, to drive exponential innovation in connectomics, one lab or few labs is not enough. We have to, I think, mobilize people, as many people as we can. So people who are going to be playing the game, people who are designing the games, right? We need to get the game industry into this. We need to get educators into this, because they'll probably want to use it for put educational content on there, which gives people another incentive to come, et cetera, et cetera, et cetera. All right, so that's enough about the means. So now let's talk about why we should care. And for that, all right, so let me just then give a short outline of my book. So the first part of my book is called Does Size Matter? And it's a short review of what we know from the regional approach to the brain. So starting in the 19th century, what people did was they divided the brain into regions, and they assigned functions to each region. Like Broca's region is for language. The occipital cortex is for vision, et cetera, et cetera. And that's still done today. That's basically what MRI is for. MRI, it gives us much more capability to do localization. But in the end, what people really care about is individual differences. So if I got 2 people, how can I explain why 2 people are different? Why are the minds of people different? And the answers that we get from studying the brain regions are not very satisfying. What are they? Well, they're things like IQ is weakly correlated with brain size. That's been conclusively demonstrated through MRI. On average, people with bigger brains have higher IQs. But if you tried to predict a single person's IQ by the size of their head, you'd be woefully inaccurate, because these are statistical statements good for large populations. And you may poo-poo that. You may say, well, actually, that's really archaic. Nobody does that anymore. But in fact, it's still done in autism research. One of the big deal finding in autism research is that the heads and brains of autistic kids are on average larger than those of typical kids. That's the kind of state that our neuroscience is in. That's all we can say. And that's a very limited statement, because if you took any kid with a big head and said they were autistic, you'd be woefully wrong. Because it's one of these statistical statements. Right? So all professional, players of American professional football are bigger on average than typical people. But if I took every big person in America and said they were a professional football player, I'd be woefully inaccurate. Okay, so the same thing with not only individual differences between people, but differences in a person over time. Suppose I learn a new skill. Neuroscience does not have any answer. It's a satisfactory answer about what's different in my brain after I learn something new, after I've changed. What happens when I store a memory? What happens when I learn how to play tennis? And so on. Again, there's experiments which show that if you learn juggling for a month, there's a particular part of your brain that thickens, on average. And so I would argue that we've got to actually figure out how the brain is organized. We have to look at the substructure of regions to figure out how it actually works. Okay, and that's the connectome. The connectome is about the organization. So part 2 is about connectionism. Connectionism is the old doctrine that indeed the organization of connections in the brain is crucial to its function. And it's been developed in all kinds of theories of neural networks in the last 50 years, mathematical theories of neural networks. But the basic ideas still it's not clear. So I talk about perception. I talk about memory, the idea that memories are encoded in your connectome. And I talk about, so mainly it's perception and memory in that chapter on connectionism. But overall, the whole idea that experiences can influence your connectome is explained there. So the next part is about genetic influences on your connectome. So there's a lot of evidence that genes have a powerful effect on minds, right? You guys know the twin studies, right? So the correlation coefficient between IQ of identical twins is something like 0.8. It's pretty high. So how do we explain genetic influences on minds? Well, one obvious way is that genes influence the connectome. So neurons have to be, during brain development, neurons have to be born, 100 billion neurons. They have to migrate to the proper places. They have to extend their branches and make connections between the branches. And all those processes are controlled by genes. So selling point for the connectome is that that's where nature meets nurture, that both influences are powerful. Then the next part of the book is about the technologies for finding connectomes and possible applications of them to looking for trying to read memories. So can you actually, suppose we had a connectome, could we actually read out the memories that were stored in that animal or person's brain? Can we find connectopathies? So miswirings. There's certain brain disorders where neurons degenerate and die, like Alzheimer's, but there's plenty of disorders in which the neurons look healthy. So the hypothesis has been for 100 years that these disorders involve wiring up healthy neurons in an abnormal way, which has never been detectable. So schizophrenia, autism, these things could all be connectopathies. It's not my hypothesis. It's already out there in the literature. And so the challenge is, can we detect those miswirings? Then the final, and then if we found those connectopathies, how could we use them to develop different therapies? And how could we use them to develop interventions that could enhance our learning, so make us better than normal? Then the final part of the book is called Beyond Humanity, and examines two kinds of transhumanism, two kinds of movements within transhumanism. One of them is cryonics. So the people who cryopreserve their brains in the hope that they'll get resurrected later on. And so I argue that to do some science here, we need to investigate whether the connectome is still intact in those cryopreserved brains. Because if it's true that your memories are stored in your connectome, and the damage to the brain induced by dying and cryopreservation actually degrades the information in your connectome, it could be that all the memories are going to be gone. So up to now, a lot of the debate about cryonics is just based on biases and preconceptions and so on. But can we actually have some science in this? And I think connectomics is one way. And then the final chapter is called Save As. So it's about the dream of uploading. Because pretty much the only plausible scheme I've heard of, remotely plausible scheme for uploading, is that of taking someone's connectome and simulating their neural network on a computer. And I discuss what's the possibility that that's going to work. You know, is the information in the connectome truly sufficient? It might be necessary, but is it really sufficient for a brain simulation? And the slogan in my TED Talk was I had people chant, "I am more than my genes, I am my connectome." Sort of communicating, it's really inappropriate because you shouldn't do science with slogans, but it's really a hypothesis, right? That your uniqueness as a person, your memories, your personality, your intellectual abilities, all those things are actually encoded in your connectome. That's summarized by the claim, "I am my connectome." So if you believe I am my connectome, then simulating, then uploading by connectome-based simulation seems like a sensible approach. But then it also causes you to re-examine this belief that that's actually sufficient for capturing human individuality. Okay, so that is a brief summary of the book. And so why don't we just do questions?
All right. Thank you.
Okay.